Right. Hello, everyone. Thank you for being with us today. Right. So just a couple of words about what our lab does. We do study bacteria and their collective behavior down to a single cell. And we opened about a year ago. We've just had our first anniversary. And you can learn more about what the lab does on our website that's listed right here. So during today's talk, I'm just going to introduce one of the research topics we're very interested in our lab and introduce this concept of a bacterial biofilm, what a biofilm is and why should we study them, what are the current challenges in studying this curious life forms of bacteria, and our technology that we've developed to study them, and how are we going to use it in the future. So in the beginning, let's talk about biofilms, what they are. So if I ask you to imagine bacteria, right? I think most people would probably imagine some unicellular organisms like this, looking kind of simple, maybe crawling around. And so this is sort of true, right? The bacteria can lead fully unicellular lifestyles. And then can grow and divide and give rise to progeny. And in that sense, each of the cells would compete against each other for survival. But then this is only a part of the story. Bacterial life cycle is actually more complicated. And it also includes sessile forms of bacterial life or adherent forms. So when bacteria have a suitable substrate, shown here in gray, they can actually adhere to that substrate and then aggregate there and signal to each other, right? And once they start signaling, they work on creating this sort of little protective bubble around themselves, which is something that's called an extracellular matrix or extracellular polymeric substances, or EPS. And it's shown here in blue. And so you can kind of imagine it as a very viscous glue-like substance that's also forming this protective shell around the cells. And so when the cells are surrounded with it, they are very well insulated from any threats from the maybe internal environment. And then the cells can grow inside this bubble until they form this collective inside this protective bubble, and this is the stage which we actually call a biofilm. And here in this biofilm life stage, the behavior of cells now is not only competitive against each other, but the cells cooperate to maintain this sort of city of microbes. They can signal to each other, and they can introduce some labor division strategies to maintain this community and help protect against external threats. And then finally, the biofilm can disperse once the cells sense that there is no more benefit to staying adherent. And so one of the examples why the cells would actually want to do this, form this community, which is very easy to imagine and is known to everybody, familiar to everybody, is dental plaque. So imagine bacteria in your mouth. Unless they adhere very tightly to your teeth, they're going to get swallowed, right? But the environment in the mouth is actually very nice for bacteria if they can adhere to the teeth, because then they can get nutrients that can be in your mouth. And so this is why the dental plaque is an example of a polymicrobial biofilm, right? Which is made of different types of microbes. Right, so pathogenic bacteria form biofilms within the body, right? Why do we want to study biofilms? Is because 80% of chronic bacterial infections can be associated with bacterial biofilms. And I'm just showing a list here to the right. It's not an exhaustive list, right? But you can see there's so many chronic or recurrent conditions that are associated with the formation of this collective bacterial community within the body. And so I think one of the most famous biofilm infections are the implant-associated infections, where you have a foreign body inside an organism, right? And so bacteria have this artificial surface to adhere, not necessarily even inside the body. It can be a contact lens. But once the bacteria colonize, for example, an implant, it can be extremely hard to eradicate that infection with an antimicrobial drug, such as an antibiotic. And so actually a very common outcome is you would have to remove the implant completely in order to get rid of that infection. So biofilm-associated infections biofilms are really a hard thing to eliminate, and they lead to very aggravated outcomes for diseases. And so why are biofilms so resistant to drugs and host defenses? So one of the examples of this resistance is shown here. So in this picture that you see an image of a Pseudomonas aeruginosa biofilm. This is an opportunistic pathogen that is responsible for infections in lungs and chronic wounds, for example. And so in this picture, you see bacteria formed a biofilm, and live bacteria are stained with a green dye, and dead bacteria are stained with a red dye. And so you can see that if we don't treat the biofilm, all of the bacteria are mostly live, but when we treat it with 3 different antibiotics, which are actually quite commonly used in the clinic to treat this type of infection, we see that some of the cells are dead, but then there's also a lot of the green cells still left, and they're located in different spatial locations across the biofilm for different drugs. None of them can actually eliminate the infection completely. So in order for us to answer this question, how is the biofilm so resistant? First, let's see how does the biofilm actually look like, right? So here in this schematic, it is shown as sort of this shapeless blob. But actually, if we look at it, for example, under a microscope, it can look quite interesting because it has spatial structure. So the Pseudomonas aeruginosa biofilm I've shown you right now actually forms this sort of beautiful spatially organized structure, looking a little bit like a flower with the center, and then the wrinkles growing radially out towards the edge. And these wrinkles actually have a functional importance because they serve as channels for water. So the bacteria obviously are doing something different in different parts of the biofilm, right? For example, if we consider bacteria at the edge versus bacteria in the center, they seem to be doing something differently because this is a very spatially organized community. Similarly, in an example of pellicle, which is a biofilm that forms on the interface of water and air, we see this interesting wrinkly structure formed by another species of microbe, Bacillus subtilis, which I'm actually mostly studying in the lab right now. So how do individual cells come together to form this complex architecture? And how can this be connected to the biofilm resilience? Right, so we know that cells in a multicellular organism share the same DNA, but perform different tasks. So for example, in our own body, right, we have so many types of cells that perform specialized functions, and a neuron is completely different in function than, for example, a muscle cell, but they all have the same DNA. So the cells just differentiate, they express different sets of genes, or they activate different sets of genes, and that makes them special and unique. So it turns out that cells within a bacterial biofilm, when they start signaling to each other and form this collective behavior, they also differentiate to benefit the whole community, right? Some portion of the cells within this biofilm can, for example, express flagella, and this makes them motile and then can swim away. Some other cells are actually responsible for producing this extracellular matrix I've told you about. And then some other cells may produce, for example, toxins, which serve to ward off the competitor species and protect their feeding niche. Some other cells may also form spores, which are very environmentally resistant life forms for bacteria. And spores are actually located in very specific spatial locations throughout the biofilm. So the cells must actually coordinate this behavior between themselves. And this also represents one of the very interesting conceptual leaps from being unicellular organism, which sort of everybody for themselves, to this collective type of behavior where the cells can perform altruistic behavior or specialize to help the whole community survive. So now, while I've introduced what the biofilm is, let's see what are the challenges in studying biofilms. Because obviously, unless we study how the cells can build the biofilms, it is very hard to eradicate them. So in order to learn what are the different cell types, bacterial cell types in this biofilm community, right? We need to measure bacterial gene expression. And so I think the most commonly used assays for studying gene expression in bacteria are what's called bulk assays. And so imagine we take all of the cells from these biofilms, right? And so they may represent different cell types, shown here on the left. And then we sort of put them in a blender and we can blend those cells and extract their RNA, which are the active form of their DNA, so to speak. And then we can maybe see what kind of genes are expressed within this community. And we can see what kind of active genes are there. But these assays are very limited in terms of information we can get out, because this doesn't tell us which cell types were there, how many of each, and also where they were. Another strategy to study biofilms would be to measure individual genes in single cells, right? So ideally, we want to take those different single cell types and count how many were there and which ones were they. And so one of the strategies that's used for that is tagging the genes with fluorescent proteins, which are the proteins that can emit different colors of fluorescence light under the microscope. And so, for example, what we can do is take the gene that makes the flagella, let's say, right, this HAG gene, and couple it to a red fluorescent protein. And then we take, for example, the gene that we know is responsible for the production of one of the components for the extracellular matrix and couple it to the green fluorescent reporter and so on. And then we can also, for example, see the sporulation gene coupled to a blue or cyan here, CFP, cyan fluorescent protein. And so then we can put this biofilm under the microscope. And this is, for example, the picture that we would see where we have this matrix. We now see where these genes are actually actively expressed within the biofilm. The matrix genes would be expressed on the outer edge of this biofilm, and then the sporulation genes would be actually expressed along these channels or wrinkles in the biofilm, and also largely in the center, which is sort of the most protected part of the biofilm. And then we have the motile cells swimming in and out through these channels, which is pretty cool. And then we can sort of merge all the colors and see what the biofilm structure is and how the cells at different locations are performing different functions. This research is not from our lab, but I'm not sure where the link to that study is. I can probably post it later. So how else can we study single cell behavior in bacteria? Right, you can see the limitations to study of biofilm with fluorescent reporters. I've only listed 3 genes that we can tag with these 3 different colors of fluorescent proteins, right? And we also need to know which ones we're looking at. Without knowing which genes to measure, we actually don't know which reporter of fluorescent protein, which gene we should tag with this reporter protein, right? Another limitation is that we cannot study the samples that are obtained from the patient or from a clinical study, right? Because we cannot actually introduce artificially any fluorescent proteins in them. So what do we do then? And this leads me to the introduction of our new technology that we've developed. I've developed during my postdoc with Georg Silik and Leandra Bretner at the UW. And so we adapted a technology that was initially used for studying gene expression on cell types in mammalian cells, which is called single-cell RNA sequencing. You've probably heard about it. It's very popular these days. So this technique doesn't need any genetic engineering of the sample, and it can also read out all of the gene's activity, which means that it can sequence or count all of the RNA that are within any single cell. And we don't need to actually pick which genes we want to measure. And so in mammalian cells, so for example, for the human brain, we can see a lot of the different kinds of neurons within the brain and so on with this technology. But this technology did not exist for bacteria. So we had to engineer it specifically so that it works for bacteria in our hands. And so once it works, we can work with clinical isolate. We don't need to engineer reporters, and we can also measure all genes. And so in theory, we could see all of the cell types that exist within the bacterial biofilm, for example, or maybe some other bacterial sample. It doesn't have to be biofilm. And we don't need any prior information for this study. And so a while back, I made this very sort of lighthearted movie about explaining this technology. And this technology, I've made it for the technology that studies the mammalian brain, but let's just imagine that it talks about the bacterial community. And not sure if you can hear the music in the video. So let's imagine that this here is actually not the brain, but the biofilm. What we start with is dispersing the biofilm into single cells represented by this ping pong ball. And then to learn the identity of the cells, we lyse them open. And then we would like to identify all of the RNA molecules in each cell. So how can we uniquely label those molecules in each cell? We put a short DNA sequence, which we call a DNA barcode, into each of those buckets. And we have lots and lots of buckets with different DNA barcodes. Here they're represented by color. They're not actually colored. They just have a different DNA sequence. And then we distribute all of the single cells into these buckets, or in our case, it's a 96-well plate. The RNA in each bucket get labeled with a bucket-specific barcode. And then we can pull them back and mix them, and then repeat this procedure using a second set of barcodes, where the RNA inside each cell is labeled by a second barcode, which is appended to the first. So like right in this purple bucket, we see the second barcode is purple. Then we can repeat this procedure for the third round of barcoding. And now, after the final split pull round, we have 3 barcodes combination. And now you can notice that each cell now has taken the unique path through the containers and has acquired a unique combination of the 3 barcodes. So now if I break the cells and isolate the RNA molecules, we can sequence these molecules all together, but then group them easily by the barcode. And now we know what the transcriptome of each cell was by knowing the identity of the cellular barcode. And so we've mapped the brain architecture, cell type architecture with this, but we can also use this to map all of the cell types within the biofilm. And so the original technology was called SplitSeq, and our microbial technology is called MicroSplit, which stands for Microbial Split Pool Ligation Transcriptomics. And like I previously said, it was developed in Georg Silik group at UW together with my awesome collaborators, Leandra Bretner and Luana Paleologu, and in a collaboration with Wilde Polo Lab, and published right here. This is the link to the paper. Right. So now how does our data look like that we can produce on bacteria? So originally we developed this technology on Bacillus subtilis and Escherichia coli, which are the model gram-positive and gram-negative species. And here I'm showing you the data for Bacillus subtilis cells that was grown in a flask from sort of low density to high density. And the OD here stands for optical density. And so you can see we have 10 samples ranging from OD 0.5 to OD 6. And how you should read this is that at OD 0.5, the lowest OD, there's very few cells still in the flask, and there's a lot of nutrients, and the cells are growing rapidly, and they're very happy. And then the cells sort of, as they grow and they deplete the nutrients, they enter something that's called stationary phase, which is where the cells basically deplete the nutrients, and then they grow to a high density. And this is where a lot of interesting behavior starts generally for bacterial cells, because now in very limiting conditions, they may implement different strategies for survival. And so the different colors here represent, in this plot, represent these different samples. And in this plot, you can see here a lot of dots, and each dot represents a cell, a bacterial cell. And so how you can read this plot is it's basically a clustering output, where if you see two dots close to each other, that means their behavior is very similar, right? So they do similar things, or they express similar genes. If you see them very far apart, like for example, this sample and this sample, that means the cells are doing some very different things. They're expressing different subsets of the RNA. And so you can see that this data, which shows some 25,000 Bacillus subtilis cells, clusters neatly according to their optical density, which is shown again as color. But then we also, and that makes sense, right? So all of the cells that are grown at OD 0.5 behave somewhat in the same way, but then as they grow, they undergo different transitions, and they're still all very similar within a single sample. But we can see some very interesting things here, where we see this cluster that is very separate from all of the cells here in the same sample, right? So if we look at the sample at optical density 2, then only about 10 to 15% of all of the cells within this cluster, within this sample, fall in this cluster. So which means this cell, 10 to 15% of all cells in the sample, do something very dramatically different from all of the other cells in the sample. So how we can actually denote that is by coloring this by cluster. And so we can see the cells form a separate cluster, which for us been numbered as cluster 9. And now we can see what the cells are doing within each of the clusters, right? And so here's this cluster 9 highlighted. So what else can we look at here? So we see a lot of central carbon metabolism changes as the cells grow and explore food sources. And so how you can read this plot, again, this data is pretty dense, but I'll just explain you step by step what you should look at. So on the y-axis vertically, you see the cluster number, and the clusters are numbered roughly according to the growth stage. So for example, clusters 0, 1, 2 corresponds to earlier growth stages on earlier ODs. And then the genes are shown on the bottom. And so wherever you see a lighter color, like here, which that means this particular gene, for example, PTSG, is mostly expressed in cluster 0 and not, relatively speaking, in other clusters, right? Or this gene MTLA is enriched in this cluster 0 and not in this other clusters. And so now we can sort of see what kind of genes are most enriched in which cluster. And so this whole figure now makes sense because, for example, right? Like I told you, the cells in the very beginning of growth, there's not that many cells, but there's lots of nutrients still present in the media. And so we can see these bacteria just happily eating the preferred sugars that they like, for example, glucose, mannitol, glycerol, and so on. But then they deplete these sugars, and as the growth stages increase, they grow more dense. They start eating other nutrients, such as mannose, for example, or trehalose. And we can study a lot of this carbon metabolism changes in the single experiment. And also what we can notice is that this cluster 9 that I've shown you just previously, which contains between 10 to 15 in general percent of cells from 3 different samples at the intermediate growth stages. These cells have turned on the genes for metabolism of inositol. And we still don't know. We've verified this with fluorescence microscopy, but we still don't know. It's still a mystery to us. Why would they turn on this inositol consumption genes in only 10% of their population? We didn't even know that inositol was present in the growth media. It's a complex growth media. But it is a very interesting observation that a novel observation that we otherwise would not be able to make if we were to not use a single cell method, because only 10% of the cells do this. And so if you just blend all of the population together, you will not see the signal for these genes. Okay, so if we switch gears from metabolism to rare behavior, which is what I'm most interested in, since a lot of the biofilm specific behaviors tend to come from sort of this rare cells. When we look at this plot, we can immediately notice that one of the clusters, or cluster 13, is very tiny. It only has 36 cells, which represents about 0.1% of the whole population. So what are these cells doing? This cluster is, however, very, very different from all the other clusters. So they must be doing something very unique. And so it turns out, here you can see this cluster that we renamed PBSX, that it does express a whole cassette of genes, about 20 of them. And a lot of them have this letter X in the beginning of their name, which basically shows you that they belong to this what's called an operon, or functional unit of PBSX prophage. And what this PBSX prophage is that an interesting ancestral phage that resides in the genome of Bacillus subtilis, and it's getting activated by DNA damage. So for example, if you shine UV light on bacteria, or in this case, this 0.1% of cells experience some random damage from just rapid growth in a normal culture. Then this element sort of wakes up. They starts making those genes, starting with an X from XPF and XRE operons, and that makes this PBSX prophage particle that sort of lyses the cell, leaks out, and kills the neighboring cells in an interesting suicide mechanism to get rid of those DNA damaged cells. And so the fact that we were able to detect this very rare behavior sort of really is very encouraging for us, because now we can use this technology to look for more rare behaviors, in particular for persistent cells, which are very rare cells within the biofilm that give rise to antibiotic resistance. And so what we will be using microsplit for next? So the data that I've shown you right now, we've obtained for just planktonic culture in order to validate and develop our technology. But now we are working on using this technology for studying the single cell behavior in a Pseudomonas aeruginosa biofilm, the same biofilm I've shown you earlier that forms this interesting structure. And it's also a very medically relevant pathogen that is responsible for the lung colonization of cystic fibrosis patients, as well as chronic colonization of deep wounds. And we do have some very preliminary data on the Pseudomonas biofilms, where we used our technology on both the biofilm cells as well as the planktonic runoff samples. And we do see it does form lots of different clusters. And these clusters have different gene expression profiles. And we're only starting to actually sort of delve deeper and to learn what all those different cells within each cluster are doing. And so I think there's lots of promise here, but we also would like to eventually use this technology not only for monospecies Pseudomonas biofilms, but for polymicrobial biofilms, because actually in patients or in nature, bacteria typically don't grow alone, right? There's almost no such thing as like a monoculture biofilm. Typically, it is several different species that mingle together to form a cross-species community. And they can not only compete in the polymicrobial biofilm, but also help each other and be mutually beneficial to form this cross-species community. And this would be an example where we have like a city that is populated by many diverse people, right? As opposed to some very similar kinds of people. So I think this is this would be a very exciting use of our technology to study how is the polymicrobial community or polymicrobial biofilm is organized. And that's pretty much it for my talk. I'd like to acknowledge my lab, Carl Geisser and Sophie Skloss, who work on making this all a reality. And also our collaborators, Georg Silik, Will DePolo, and my awesome co-authors of the microsplit technology, Leandra Bretner, Luana Paleologo, and many, many others who helped us and who we are collaborating with on this project. And that's it. Thank you very much.
Much, Anna. So we do have a few audience questions, and folks can feel free to add more in the Q&A at the bottom of your screen. But first question is, how can this work lead to better treatments of bacterial infections?
Right, so that's a very interesting question. So in order to treat something well, we need to understand something well. And this work is basically aimed at shedding light at some very fundamental concepts of how can bacteria transition from this very simplistic life as unicellular organisms to a biofilm or collective life cycle. And there's many signaling pathways that are activated specifically for biofilm formation, or for example, production of the extracellular matrix. An extracellular matrix is one of the very important components of the biofilm that make it extremely resistant. It's estimated that cells within a biofilm are 5,000 times more resistant to antibiotics than free-living or planktonic cells. And so there are strategies, for example, using specific chemicals or enzymes to disrupt the extracellular matrix, using nucleases or proteases that would help degrade this sort of protective layer that surrounds the biofilm, and then the antibiotics would be better penetrating. So that is basically a combination style therapy that's being proposed. There's also many new therapeutic agents that are being developed. For example, nanoparticles are now being used to treat or help treat biofilm infections. They're not used in by themselves, but typically in conjunction with antibiotics or antimicrobials. And so in order for us to come up with this new treatment ideas for combination therapies for how to make antimicrobials work better for biofilms, we need to know what kind of signaling pathways within the biofilms or cell types are very important for the biofilm, and then we can target them more specifically.
Good. Very promising. Next question. There are a lot of similarities in language between your work and cancer treatments. Why is that?
Yeah, fascinating question. I think what happens in this chronic bacterial infections when people are treated for them is sort of similar or reminiscent of what happens in cancer therapy, where the patient is given a drug and it seems like it worked and the infection has stopped, but then it relapses. And the conceptual similarity here is that if the infection has been associated with a biofilm, for example, the bacteria inside the biofilm are very differentiated, like in cancer. And so the treatment may efficiently eliminate a lot of the cells, like 90-something% of the cells, but just a few of them may remain. And in the case of the infection, that will be those persister cells, maybe, or spores, hidden within the biofilm and so protected, right? And then you will have the relapse and infection. And I think it is important. I think this infectious disease therapies and cancer therapies can sort of employ similar conceptual strategies to deal with this type of persister cells by using combination therapies or priming the immune cells to learn which kind of cells to attack.
Interesting, thanks. And another question, what would be the next technological advance or innovation for studies of biofilms, or what might be?
Right. So in the technology that I've just introduced in microsplit and in SplitSeq, we do need as a first step to dissociate the biofilm, right? And so we will have a single cell suspension, and now we can study what kind of cell types are there on a single cell level. But we can't, because we lose the spatial structure, now we can't very easily map them back to where they were originally located. But there are technologies for mammalian cells right now that can simultaneously measure. They can introduce sort of spatial tags or a spatial map using a different kind of DNA barcoding. And then they can dissociate the tissue, for example, but they will still retain the spatial address of where the cell initially was. And so this is something that I think would be very exciting to bring to this technology, where we have where we would have an idea of, for example, which cell types were together and maybe communicating, or which ones were in different parts of the biofilm.
Thank you. And another question. We hear how antimicrobial resistance is a major problem. How might your research tackle AMR, if at all?
Right. So antimicrobial resistance is a very broad term. And there is many types of antimicrobial resistance one can refer to. So one major type is the genetic resistance, where the bacteria accumulate mutations that makes them resistant to antimicrobials. What we are dealing here is our technology can look at is another type of resistance that actually I think gets quite less attention, but is also very important, which is called phenotypic persistence. And in this type of persistence, the cells don't mutate. Their DNA is the same, but they change their gene expression profile. One example of that would be going dormant. So the cells may be just metabolically dormant and not growing at all. So if you use antibiotics that target actively growing cells, those will be spared. But then they can actually switch back from the state of metabolically dormant persisters to actively growing cells, and then they can repopulate the whole population without ever needing a genetic mutation for any of this. It would be just a switch in behavior, so to speak. And we are hopeful that we can find those persister cells within this larger communities, because we can do single cell resolution studies. And there's been indications that this persister cells come in different flavors, so we're very excited to see which kinds we can find.
Yeah, that could have a huge impact. So another question, you mentioned single cell sequencing. Do you think metabolomic, I might be mispronouncing that, metabolomic could help, or can we do single metabolomic for the bacteria? And correct my pronunciation if I was metabolomic.
Metabolomics. Okay. Right. I'm not very well versed with metabolomics. I think we're quite a ways away from doing single cell metabolomics, at least in the same scale as single cell RNA-seq. So single cell RNA-seq is pretty advanced now that you can do, for example, for our methods, you can easily profile hundreds of thousands of single cells in an experiment pretty easily. Metabolomics for single cells, I feel like that would be harder, especially for bacteria. But I think one day it could be possible.
How might this method be adapted to diverse microbial communities like those in the human gut? What are the challenges there?
Yes, thank you. A very fascinating question. And like I mentioned in the introduction, biofilms are only sort of one of the research topics we're interested in. The other research topics we're pursuing is studying the microbiome or microbiota. And this method is, I think, holds promise for studies of diverse communities, diverse bacterial communities, because it is sort of genome agnostic. You just sequence what's out there. You don't really need to synthesize specific probes for a specific type of bacteria that you're looking at. I think the challenges here would be permeabilization, because the bacteria are so diverse. And we do need to get those probes inside the fixed and permeabilized cells in order to label those RNA molecules. And so you can imagine that one kind of permeabilization treatment may work on some types of cells and maybe not as well as some other types. Surprisingly, though, the treatment we came up with works on two major types of cells, gram-positives and gram-negatives. And it's the same treatment, it just works on both. So we are quite surprised by it, but it's also a very promising sign that we may be able to find a treatment that works quite well for at least a large portion of, for example, human gut microbiota species.
Great, thank you. Another question, sepsis. How does your work advance this study of this common infection and fatalities in hospitals?
In hospitals. So the hospital-acquired infections, as far as I know, oftentimes are these very tough cases of multiple drug-resistant bacteria. And so I don't think there's much known about how the bacteria become multiple drug-resistant. But I think this is specifically the case where we desperately need new ideas or new targets and how to treat those infections, besides just using the same antimicrobials. And we also, I'm not sure if, there is much known about the persister cells within those strains. So I think looking at that would be very interesting. Also, the same infections also form biofilms. For example, one of this notoriously nosocomial infection or hospital-acquired infection is the multiple drug-resistant Staph aureus, which is extremely hard to eradicate, very commonly acquired in hospital, and it does form biofilms. So that's all sort of related to my research topic.
Great. And one last question, do you have a favorite bacteria that you study and why, if you do?
It's a fun question. I don't really. I want to say no. I'm kind of attached to Bacillus subtilis, which is actually not a pathogen, but a soil bacterium. It's a harmless, there are probiotic in even edible strains of this. If you've heard of Bacillus natto, which is a probiotic food from Japan, makes this natto bars. Anyways, so this bacterium is actually a nice model system for us because it does form biofilms very robustly. And it also manifests a lot of different behaviors. It forms spores, it becomes motile. There is actually a programmed cell death within Bacillus subtilis biofilm. So it's a very fascinating bacterium that has lots of trick up its sleeve, so to speak. And it's also a very convenient model system that I've used and studied for many years. So I'm kind of attached to that one. So it's a very good model system, but we will be certainly moving to pathogen models in the future, because I'm interested also not only in developing these technologies, right, using harmless models, but using it to advance our knowledge of pathogens.
Great. Well, thank you so much, Anna, for sharing your expertise with us today, and we'll be following along as you continue this line of inquiry. And thank you to everyone in the audience for being here. We'd love to see you at a future event. For those in the Seattle area, please save the evening of April 6th, 2023 for a talk at Town Hall Seattle with Drs. Leroy Hood and Nathan Price about their soon-to-be released book, The Age of Scientific Wellness: Why the Future of Medicine is Personalized, Predictive, Data-Rich, and in your hands. If you're on our email list, you'll get more information about that. And you can also go directly to our ISB website and find information under events. Those who are interested in learning more about systems biology may also want to tune in to the January 20th episode of the Technically Human podcast. That's called Science for the 21st Century: Understanding Systems Biology, and features ISB Senior Research Scientist Dr. Jared Roach. And if you have ideas for topics for these research roundtables, please contact us via the website. We'd love to hear your ideas. The series is aimed for non-scientists, so we can, get this work out and make it approachable for all. And we appreciate feedback, and we're grateful that you were with us today. Have a good evening.